Okay. So we left off last time, which was a full week ago, I think, talking about sex-linked inheritance or sex-linked genes. And I just want to remind you, our sex-linked genes, at least for mammals and for Drosophila, these are genes that are found on the X chromosome. And the reason why we call them sex-linked is because male and female mammals and male and female Drosophila have a different number of X chromosomes, right? Typically, male mammals have one X chromosome, whereas females have two X chromosomes. And so what that means is for any of those genes, our recessive character is going to be more common in males than it is in females. Because basically males only have one copy of the X chromosome. So even if they only have one copy of the recessive allele, they are going to express that because they only have one copy. Whereas females, they have two X chromosomes. So if they have one recessive allele, as long as they have one dominant allele, they are not going to express the recessive form of that character. Now, the opposite is true for dominant characters. So if you've got a sex-linked dominant genetic illness, that is the gene that is related to that genetic illness is on the X chromosome, and the dominant form of that allele triggers the genetic illness, females are going to be more likely to have that because they have two opportunities to get one dominant allele, whereas the males only have one opportunity to get a dominant allele. And yeah, those genetic illnesses are fairly rare, but there are some. I want to say phosphatemia. Hypophosphatemia is one of them, but I don't know that for sure. And then, so this is a question we talked about a week ago, but I want to ask this question again. What do female mammals and female Drosophila do about having an extra copy of each of those genes on the X chromosome? What do? Does anyone remember? It was a whole week ago and a lot has happened.
They shut it down.
They shut down one of the X chromosomes. And that's why you see like calico cats, the gene for the pigment, brown or orange pigment, it's on the X chromosome. And so depending on which one they shut down, okay, there's going to be a specific color expressed, either brown or orange. Yeah.
And it's cell, it depends cell to cell. One may shut off one.
Yes. Yeah, but now what tends to happen is that shutdown process happens early in development, and then all of the cells that descend from that cell that shut down the same X chromosome. Yeah. And so that's why you have huge patches of orange and huge patches of brown, and if it was individual cells, it should look like a mosaic, and so for far away it should just look brown, right? But it's because that the shutdown process happens early in development, as does the mutation that results in the inability to deposit pigment. And that's why you get big white patches as well, because that's a separate mutation that makes them incapable of depositing any pigment. Whether it's brown or orange doesn't matter. They're not depositing it. Yeah, Micah.
Mutation that stops the deposit.
Yes.
Is that related to shutting down the X chromosome?
Epistatically. It would be an epistatic effect where you would say no pigment is epistatic to orange and brown pigment. Same like what we had with our Labradors, where that yellow, the yellow coloration is the result of an epistatic gene that prevents pigment from depositing. So you can't deposit the black or the brown pigment. It just is no pigment and looks yellow.
So are there cases then when you have brown with orange splotches?
I mean, the only way to have that would be to have something happen and it shift which X chromosome was getting shut down after, and that very rarely happens. Is it possible? Yes, but it very rarely happens. Okay. Yeah, Autumn.
Does that affect them in any way other than the different colored pigment?
You mean does that result in, I mean, the darker the pigment, the more sunlight they're going to absorb there, right? And so the more likely they are to, in the lighter parts of their body, they're more likely to have UV light penetrate and to, one, facilitate more vitamin D production, but then also to result in higher amounts of mutations. And so they're more likely to develop skin cancer in an area where they have lighter pigment or no pigment than in the places where they have darker pigment. Yeah. And then I'm sure there are other implications as well, that the darker pigment parts of their body will be warmer when they're in the sunlight than the other parts. So what does that mean for the cat? I don't know. All right. So I told you that this chapter it's a little bit of an interesting one because we have to talk about what are all of the other genetic processes that influence human genetics, that explain why human genetics rarely conforms to Mendelian predictions, right? And so a week ago, we talked about sex-linked inheritance, right? That in humans and in all mammals and in Drosophila, sex is determined by the presence or absence of a Y chromosome. And then so that changes some of our predicted ratios if the gene is on the X chromosome. Another thing that influences this is in human genetics, we have a lot of evidence of chromosomal aberrations, of changes in chromosomal number. So when they initially published the findings of the Human Genome Project, this was back in the early 2000s. 2002, I think, is when you got the whole suite of papers that came out. Maybe it was in 2001, and then they officially put the project to a close in 2002. But anyways, when the initial publications came out, the only way that they were comparing one human genome to another was in what you would call single nucleotide polymorphisms, where you had a single place in the genome where most people say had an A, but some people had a G, right? And so it's single nucleotide polymorphisms, one place where you can differ. And from those, they said that humans collectively only differ in 0.1% of our markers. So basically, they as part of the Human Genome Project, they published that humans are 99.9% identical genetically. The problem is they weren't looking at chromosomal changes, where you actually had pieces of chromosomes missing, because the assumption was that that was a very minor part, but it's not. It actually accounts for 4 to 5 times more variation than single nucleotide polymorphisms, having a single base that instead of being an A is a G. Okay? When you're actually looking at that, people are missing parts of chromosomes. And so these are called copy number variants, and they account for about another 0.5% of variation. So almost all humans vary by about 0.5% of our genome. Now you've got some like really reproductively isolated groups that I don't know how, but some geneticists have gotten some genetic samples from and compared their DNA, and some of those groups are actually pretty dissimilar. So some of the Aboriginal groups in the Oceanic Islands, some very remote tribes in South America and in Africa, that they may vary, their genomes might vary by up to 5% from other humans, and it's because of reproductive isolation. But on the whole, humans vary about 0.5% of our genome, and most of that variation is in chromosomal mutations, changes in number. So we need to talk about chromosomal mutations. Here, our first example of a chromosomal mutation, we call this a deletion. And why? What happened from here to here? Here's the norm, or what we might call the wild type, and then here's the mutant. And what's missing? The F. It got deleted. That's why we call it a deletion. Isn't that beautiful? The F, that portion of the chromosome. So whatever was here, and if you look at the chromosome, it's not the letter F, right? There's only A, T, C's, and G's. It's just this region of the chromosome was deleted over here. And this happens a lot. It happens a lot. And we know this happens a lot because if you were to compare my genome with your genome, you'll find regions that you have in your genome that I don't have in mine. And you'll find regions that I have in my genome that you don't have in yours. And you're like, that's so weird. And it is. It is. All right. And then here we call this one a duplication. And so here's wild type. And then here's the mutant. And what's different? DE was duplicated. And that's why we call it a duplication. Isn't that wonderful? Because like we could totally call this anything we want, but whoever discovered this called it what it actually is. And that's convenient, because scientists rarely do that. You know, typically what a scientist would do when they discover this, they'd name like either the biochemical mechanism of what's happening, they'd name it that, or they'd name it after somebody they don't like, right? It's like, well, how do we name this mutation? We'll call it Beth, right? Like, it's so weird. Why would you call it that? It happens a lot with parasites. When you look at the names of parasites, you're like, where did this name come from? And then you learn the person that first described the parasite, and then you study a little bit about their life, and you're like, man, I totally get it. I totally get it. They named this parasite after their dad. Totally get it. Yeah. All right. I would never name a parasite after my dad. Maybe my brother, but never my dad. All right. This one getting a little bit more complex in the name, reciprocal translocation. And so reciprocal means basically that it's give and take, right? It's reciprocal. You know, like if somebody's like, you know, I love you, and you're like, yeah, I reciprocate. You know? You don't talk like that? You're like, it's reciprocal, you know? The feeling is mutual. Like, just say I love you, right? Or give a hug, you know? Anyway, sorry. This is another story for another time. So, reciprocal translocation, you actually have pieces of chromosomes that are exchanged. And usually this happens on non-homologous chromosomes. Because you're like, isn't this crossing over? This is good, right? We talked about this, that it increases variation. You're Dr. Engel, you told me this was wonderful. In fact, I'm pretty sure you told me it was beautiful. And now you're talking about it being a chromosomal mutation. Well, when it happens between non-homologous chromosomes, look at this. This chromosome's A through H. This chromosome's K through N. Those are different chromosomes, yes? And they even colored them differently. But some of you have to trust me on that. So, but they're lettered differently. And look, we exchanged some information. So FGH from here went to here, and N came in return. It was a reciprocal translocation. They exchanged some pieces of information. And you're like, okay, so I can understand what's going on here, but why is this bad? Why is this bad? And so something we haven't talked about yet, because we haven't done gene expression, we haven't talked a whole lot about the control of which genes get transcribed and then translated. Right? We talked about that here's a gene. The central dogma of molecular biology tells us that a gene exists to be transcribed and then translated. We know it's a little more complicated than that, because some genes get transcribed but never get translated. In fact, most genes get transcribed but never translated. And then some things get transcribed, modified in a whole bunch of different ways before they get translated. But something we didn't talk a lot about is what controls whether or not they get transcribed. We kind of just learned it, and you're like, cool, if it's a gene, it's going to get transcribed. But that's not necessarily true. In fact, for most genes in an individual cell, they don't get transcribed. They never get transcribed because there's something in that cell telling that gene not to get transcribed. And we call this the controlling of gene expression. Here's a potential issue. Let's say this region on this chromosome codes for what we would call an oncogene. And you all remember from our chapter on the cell cycle that oncogenes, those are the cyclins, right? Remember those cyclins? Like cyclin B, cyclin A, cyclin, what do we call the M cyclin? I'm giving you the actual, sorry. We talked about M cyclin and G1 cyclin and G1S cyclin, right? And S cyclin. You remember all those? Yeah. Those are what we call oncogenes. Because if you start transcribing them, you're going to move through the cell cycle whether you're ready to or not. Right? And we even saw that in that research when we took a cell that was in G2, we brought a cell that was stuck in G1, you could call it G0 even, and you brought it next to the cell in G2, what happened? That cell immediately moved to G2, and it never did S. It just immediately went to G2 because it started making the cyclin to move into those sections. And so we call those oncogenes because when they're overexpressed, they develop uncontrolled cell growth and they develop tumors. Okay? So let's just say that this right here is an oncogene is sitting there. And it's sitting in a relatively inactive place in the chromosome, being told, don't transcribe it until we're ready. But then it gets moved to here, and maybe this is a really active part of the genome, where it's always transcribing. So it gets moved here, and it's like, always transcribe. And then now something that should not be transcribed until it's ready is being transcribed all the time. And the cell's just moving through the cell cycle like crazy, and leads to uncontrolled cellular growth. And here's an example. This is a one of the mutations that leads to a development of one of our blood cancers, chronic myelogenous leukemia, and a portion of chromosome 9 is reciprocally translated with a portion of chromosome 22. And what it does is it takes one of these oncogenes that should not be transcribed, and it puts it into a place that gets transcribed like crazy. And so what it does is it creates what we call a mutant chromosome. There's a name for it. It's called the Philadelphia chromosome. Not because this increases your ability to love your brother, but I don't know why it's called the Philadelphia chromosome. I'm guessing because it was discovered somewhere in Philadelphia. I don't know. But what it does is it leads to an overexpression of these oncogenes, uncontrolled cell growth, when this blood cells are developing and they don't develop properly because they just keep replicating. They just keep replicating, and so they don't develop properly. And so you end up having white blood cells that aren't developed properly, don't behave properly. Okay? Does that make sense? It's not good. It's not good. Now, not all reciprocal translocations lead to some pathology, but they can. And just like not all deletions lead to pathology, but they can. Not all duplications lead to pathology, but they can. In fact, many of them don't and just make us different. Chris?
So, what would cause chromosomes to transfer? Because the only thing I can think of.
Yeah, they line up next to each other during prophase 1 of meiosis when they shouldn't.
Okay.
Yep.
And then it just trade the wrong.
Yes.
Okay.
And it's only going to happen if there's enough homology there that it works, but it does. And it usually happens at the ends of chromosomes, where we've got some regions where there are a lot of similarities among chromosomes. Okay, I think we just have one more. One more chromosomal mutation. This one's called an inversion. And it's so wonderful because an inversion means to turn it around, right? I took my daughter to the dentist last week, and they opened up a chest, like a toy chest. They're like, you did such a wonderful job. She's 3, and she actually did do a really good job. But they're like, you get to choose anything from the chest. You know what my daughter chose? She chose an hourglass. And it made me so proud, because I'm like, there's toys in there. And she's like, I want that. But you know an hourglass, when you invert it, right, the sand drops, and she thinks it's so awesome. They do it so it gives you like a 2-minute clock for you to brush your teeth. But she does it all the time. Like, she'll just put it on the counter, and she'll just watch it, and then when it stops, she, like, becomes an alarm clock and lets everybody know that the 2 minutes is up. It's awesome. It made me so proud, because I'm like, she's a nerd. She's a nerd, and she's 3 years old. I love it. She wants time-telling tools, right? Keep track of how much time it's taking her brother to do something. I'm just waiting for the time. Anyways, sorry, that's another story I'll tell you it anyways. So my son, he's 5, and he takes a ridiculously long amount of time to do anything, right? So you're like, how long should it take a 5-year-old to change into pajamas? I don't know, a minute at most. And he's like 12 minutes. Because he's like rolling around on the floor and trying to take off his pants without using his hands. Right? Why? Because he's 5 and he can. So I'll have to like set him a timer, and I'm like, you have 2 minutes to get this done. And our daughter hasn't connected it yet to her little clock, but eventually she's going to, and she's going to bring it to him and let him watch it. Yeah. Gosh. I'm sure I did that kind of stuff when I was 5 too, but man, it is frustrating. Also very interesting to try to watch a 5-year-old get changed without using their hands, like wiggling out of things. Fascinating. All right, so sum all this up. What are chromosomal mutations? Well, these mutations, they change the structure or the number of chromosomes. We haven't really talked yet about the change in the number. We talked about the change in structure, right? Delete a portion, duplicate a portion, invert a portion, translocate a portion. But we'll start talking about how do we change the number of chromosomes. Yeah, Chris?
Did you skip inversion? Or
No, it was the last one I did.
oh,
Sorry. Right here.
oh okay.
Yeah. Now, I described inversion, but actually didn't point this out. So here it went EFG, but then it got inverted, and now it runs GFE. And most of the time, an inversion has no impact on the chromosome. The only way it does is if basically there's a gene that writes over this boundary. If there's a gene that writes over that boundary and you invert it, you destroyed that gene, right? Other than that, they usually don't have a lot of impact. All right. How does the structure change? Well, the fragment can be lost. We see this. There a specific human mutation or a human genetic illness called cri du chat, which means the cry of the cat. It's a missing portion of chromosome 5, and the disease actually has a number of phenotypic issues. A lot of them are developmental. You get mental and developmental abnormalities, and so they make a really high-pitched squeal, and that's why it's called cri du chat, or the cry of the cat. The fragment can insert into the homologous chromosome. This is the major way in which we get a duplication. And this is an explanation that is used by evolutionary biologists to basically describe some of the major evolutionary events. What's interesting is when you look at vertebrates compared to other animals. So vertebrates, fish, amphibians, lizards, birds, mammals. When you look at vertebrates compared to the rest of animals, vertebrates are very complicated developmentally. Very complicated developmentally. There are other animals that are complicated, but vertebrates as a whole, they're very complicated developmentally. And the development is very temperamental. So like you can alter the development of a sea star or a sea squirt or an insect, and it'll still develop. It'll just be weird, right? Like you can get legs to show up where the wings should be, or wings to show up where the antennas should be, and it's fine. Like they're weird and they look like, I don't know how to say this other than they look like freaks, but they still develop. When you do that with vertebrates, they oftentimes they don't develop. It is a very highly controlled process. And so what you find is that vertebrates have way more of what we call the master control genes than other animals do. The genes that control the development. When it happens, where it happens, okay, because they regulate the expression of other genes. We'll talk about this more next week. And so the idea is that in the original evolution of vertebrates, that we had like a duplication event to duplicate those master control genes. To duplicate those master control genes, giving higher levels of control over the development. It's an interesting story, one that we'll talk about further next week. All right, then fragment can insert into a non-homologous chromosome. We call this translocation. It's usually reciprocal, causative of many cancers, where you take an oncogene and put it next to a highly transcribed region of the chromosome. For example, chronic myelogenous leukemia, CML. Or the fragment can reinsert backwards. Essentially the same effect as translocation if you draw it to a highly expressed region. And again, the idea here is this is also very important in the evolutionary process. This has been kind of abandoned. This came from the idea that, I guess it hasn't been fully abandoned. So what's interesting is another different thing about vertebrates. So vertebrates, our major nerve cord runs along our dorsal surface, runs along the back, right? This is our major nerve cord. All other animals, if they only have one major nerve cord, it runs along the ventral surface. Okay? And you're like, wow, that's really weird. And so the idea is that the gene that basically sets up the dorsal ventral axis, somebody was like, I bet it's reversed in vertebrates compared to other animals. And that way it basically is saying like this is the ventral surface when really it's the dorsal surface. And you know what's cool? When you go and you look in the vertebrate genome, it's exactly reverse from what other animals have. That the gene that establishes that axis. And like that's so cool. And we'll talk more about that next week. What does that actually mean? All right. Okay, so up to this point, we've talked about changes in chromosomal structure. We need to start talking about changes in chromosomal number. And as we do, we're going to take a little bit of a lecture break. We've been going for like 30 minutes. And so what I want you to do is this. I want you to work with those around you, and I want you to come up with any examples that you know of where we have an abnormal chromosomal number. Any examples that you know of, okay, that you've heard of, you've learned from other classes. And if you can't come up with anything, we'll talk about this collectively. So take a couple of minutes, work with those around you, and see if you can come up with any specific examples of changes in chromosomal number. 2 minutes, starting now.
You know what?
Oh yeah. Oh, is it
Like Superman syndrome where they have an extra life.
Well, what happens with that one?
You don't turn Superman, that's for sure.
Extra Y
Sex
chromosome.
chromosome, or you get like two X chromosomes and a Y chromosome. Yeah.
You get the best of both worlds at that point, yeah. You get triple.
Oh, really?
What's the...
You said he has a lot of allergies.
Well, you said
Oh, yeah.
I always thought down one.
Yeah.
I think those.
Wow, it took me 2 minutes. No, that's been 1 minute, sorry. Sorry, it's been 1 minute.
Hey, you take this one. This Superman. You gotta be taller.
Are you talking about Super Y
Is that what
or Superman syndrome? Yeah. Super Y or Superman syndrome.
What is that?
I mean, my family don't have
Usually you're sterile, but not always.
So like,
It does correlate with some mental deficiencies and with some increased aggression and increased size. But if you're not sterile, you'd be a lot more likely to produce male offspring.
Oh, right. Just not a little Y.
Yep.
Yeah.
Hi everyone. Welcome back. Did you miss me?
Yes.
Does it bother you that I leave during that time? I use it to go get water because I feel awkward waiting here for you, so I go get water. Sorry. Yeah. What are some of these issues that are involved with a change in chromosomal number? Yeah, Davies.
Down syndrome.
Down syndrome, an extra version of chromosome 21, although sometimes it's just an extra portion of chromosome 21 and not the entire chromosome. So there are multiple ways to generate Down syndrome. You could generate it from what we call aneuploidy, a change in chromosome number, or it could be a reciprocal translocation, and then, anyways. Yeah, any others? Yeah, Allison? There is. Yeah, and I forget what it's called. Edwards? No. Is it Edwards? Trisomy 18? Somebody look that up. See if Edwards syndrome is trisomy 18. Yeah.
One with two X's and a Y.
Two X's and a Y. Yeah, that's called Turner's. Oh, no, Turner's is 1X. That's Klinefelter. Edwards is trisomy 18. Yep. And then we've got two X's and a Y. That's Klinefelter syndrome. So in all mammals, Klinefelter syndrome happens in all mammals and probably in Drosophila as well. And the phenotypic implications of it vary. Because remember, sex is determined in mammals and in Drosophila by the presence or absence of the Y chromosome, not by the number of X chromosomes. So Klinefelter syndrome, some individuals would have no pathology. They'd be fully biologically male because one of those X chromosomes would get shut down just like it does in female mammals. Now, it can oftentimes lead to sterility because when you go to do meiosis, there are 3 chromosomes that line up side by side instead of 2, and it fails that checkpoint and just shuts the process down. But then you have Turner syndrome, which is 1 X chromosome, no second X chromosome. And again, the sex is determined in mammals and Drosophila by the presence or absence of a Y, not by the number of X's. If males function with only 1 X chromosome, females can function with only 1 X chromosome. And again, sometimes sometimes there's no pathology with Turner syndrome. Sometimes there is, but oftentimes they are sterile because there's no second chromosome to line up during meiosis 1, and it fails the checkpoint. And then you have super Y or Superman syndrome, where you have XYY, or sometimes even XYYY. Yeah. And then there's trisomy 13, and I forget what it's called. So there's Edwards, which is trisomy 18, and then there's trisomy 13. I think maybe it's just called trisomy 13. Is there a name for it? Patau syndrome? Patau. I should have known that. Yeah, Allison.
So is there a reason that some chromosomes are more likely to triple like that?
It's not that the chromosomes are any more likely to triple, it's that other trisomies are fatal. And it's just that some of them aren't fatal. So Down syndrome tends to be the least likely to result in fatality in the autosomal chromosomes, so it becomes the most common trisomy. It's not because it happens more often in chromosome 21, it's just the least likely to be fatal. Followed by Edwards and Patau. And then trisomy other chromosomes are always fatal. And that's why you just don't find them. It's not because they don't happen very often, it's just they don't survive. And then trisomies and other aneuploidies in the sex chromosomes tend to be handled better because our bodies are designed, mammals are designed, and Drosophila are designed to handle different numbers of X chromosomes. And so those tend to be less significant in terms of contributing to pathology. You also have what's called triple X syndrome, where you have females with 3 X chromosomes instead of 2. So then you would have 2 X chromosomes would get shut down in every cell. It varies. Anywhere from no implications to some developmental and mental abnormalities. Yeah. All right. So the most common cause of aneuploidies, which is a change in chromosomal number. So everything we talked about before is a change in chromosomal structure. Now we have a change in chromosomal number. The most common cause of aneuploidies is nondisjunction. And so nondisjunction is where the chromosomes don't separate. Either during anaphase 1, the homologous chromosomes don't separate, or during anaphase 2, the sister chromatids don't separate. In this case, we're showing it happen during meiosis 1. During anaphase 1, the homologous chromosomes didn't separate. Both versions of this chromosome got pulled over to this pole, and so all 4 of our cells have an abnormal chromosomal number. These gametes are missing a chromosome. These gametes have an extra copy of one of the chromosomes. So then when they were to go and fuse with another gamete, would have an abnormal chromosomal number. And so let's say this is chromosome 21. And so these 2 gametes would result in an extra copy of chromosome 21 in the zygote. These 2 copies would result in only 1 copy of chromosome 21 in the zygote. This would be fatal. This one can be endured, okay, but does contribute or does lead to significant developmental differences, abnormalities. And so here's a karyotype of trisomy 21 of Down syndrome, and here's a 3rd copy of chromosome 21. Most often the result of nondisjunction, and most often the result of nondisjunction in ovum formation. Okay, in mammals, as far as we know, this is true of all mammals. All of the gametes that a female mammal will have in her entire life are developed during or in utero, and then are put at an arrested state of development until they're fertilized. And when they're fertilized, they'll finish development. And so the older those cells are, the longer they've sat, the less likely the chromosomes are to segregate properly when they finish development. And so here's a graph looking at the relationship between the mother's age and incidence of Down syndrome. And again, most of these trisomies, and this is going to be true for all trisomies, okay, that there's going to be a relationship between the mother's age, and so really the age of that ovum, the age of that secondary oocyte, and the development of Down syndrome. But it's going to be true for all trisomies, because the older that secondary oocyte is, the less likely the sister chromatids are to split properly when they finish up the formation. Yeah.
So when you say that during development all the ovum are
all of the gametes, but they're not fully developed. They're not ova yet.
okay.
They actually don't even become ova until after they're fertilized. And then once their nuclei fuse together, then they're zygotes. So they're actually only ova for a very, very short period of time. Because what happens is they go through meiosis 1, and then they start meiosis 2 and they stop. They stop meiosis 2 at metaphase 2. And so they're stuck there, and they only finish it if they get fertilized by a sperm. Yeah. Okay. Any questions? So here are the nondisjunction or the result of nondisjunction in our sex chromosomes. So here we started with a cell that was XX, so this is a female germ cell that's going to give rise to gametes. And then here we had a nondisjunction event, and so you had one gamete that's got 2 X chromosomes, one gamete that has none. And so if you have your male contributes a Y chromosome here, you're going to have a Y chromosome but no X chromosome. This is not going to survive. You have no X chromosome. There are genes on there that you need. Here you have an X chromosome fuses with this gamete, so the male gave his X chromosome, fuses with this ovum, and you get XO. This is Turner syndrome. Again, the degree of pathology is going to vary from individual to individual, but it's possible to have Turner syndrome and have no noticeable effects, although you would very likely be sterile. Here, you've got XX with the Y, so now we have Klinefelter, and then here we have XX and another X from the sperm, triple X syndrome. And again, the pathology here is going to vary from case to case. So here's a table out of another textbook that shows you these chromosomal mutations, their frequency. So the most frequent is 1 in 1,000, still very rare. And then some of the effects. Apparently normal female with normal or slightly retarded mental function. Most of the time is going to be normal. And then XYY, apparently normal males, but taller than average. There is some correlation between increased aggression with, this is called Superman syndrome, XYY, and then XXY, and then Turner syndrome. Okay. Oh, so then connecting this specifically to pathology. How does aneuploidy result in pathology? Entire chromosomes, in aneuploidy, entire chromosomes are lost or gained through nondisjunction. So you either have too much or too little. We call these individuals aneuploids. Now these usually lead to miscarriages where they can't survive. Some exceptions, extra chromosome 21 with Down syndrome, probability increases. Also Edwards syndrome and Patel syndrome with chromosomes 18 and chromosomes 13. Alterations of sex chromosomes are usually tolerated. We talked about this, Turner syndrome, Klinefelter. And then if you change the entire number of chromosomes, so every single chromosome, you get an extra copy. This is almost always fatal in animals, but it's very well tolerated in plants. And so this is ways in which we've actually genetically modified plants to produce, say, higher levels of gluten, to make high gluten flours, so you can make pasta and pizza, or to make flours larger. Okay. So, I think this is the last question that we're going to deal with. What are the inheritance patterns of human characters? So let's talk about specific characters, sometimes specific genetic illnesses, and follow their specific inheritance patterns. Okay? So many of these genetic illnesses do follow Mendelian inheritance patterns, some autosomal recessive. Sickle cell disease, cystic fibrosis, phenylketonuria. These are all genetic illnesses that are autosomal recessive. So the gene is it's not on a sex chromosome, it's on chromosomes 1 through 22, and you need 2 copies of the recessive allele in order to have that genetic illness. Okay, phenylketonuria, have we talked about this? I've talked about this in genetics quite a bit. It's the inability to metabolize phenylalanine, which is one of our amino acids. And so you end up building up a toxin if you can't metabolize that. And so it's treated fairly well by just having a diet that has a very low amount of phenylalanine. So it's one of the things that they screen for in newborns. They screen for PKU because, again, it's fairly easy. You can't drink diet beverages, and then there are a couple of other things that are high in phenylalanine. So only homozygous recessive individuals have the genetic illness. So this is going to be 1 in every 4 offspring in a monohybrid cross. So even if you had 2 carriers, like say 2 people that are carriers for cystic fibrosis, on average 1 in every 4 of their offspring will have the genetic illness. Then we have autosomal dominant traits. Talked about some of these already. Achondroplasia, which is short-limb dwarfism, and progeria, which is an aging disease. You age too rapidly. Even heterozygotes have this disease, and most of the time, homozygous dominant individuals have such an extreme form of the disease, they don't survive. And so it's usually only heterozygous individuals that are going to survive and demonstrate the disease. X-linked recessive traits, red-green color blindness, hemophilia, Duchenne muscular dystrophy, disorder and how your muscles are developed, hemophilia, bleeding disease. We've talked about red-green color blindness. That one speaks for itself. So these are more common in males, and that's because males only have 1 X chromosome. So if they get 1 copy of the recessive allele, they are going to have that genetic illness. And then X-linked dominant, constitutional thrombopathy, which is an issue with your thrombocytes, which are cells that work in the clotting process, hereditary enamel hypoplasia, the inability to develop enamel, and then I'm pretty sure hypophosphatemia is one as well, but I didn't put that one there. You shouldn't either. Probably the same in males and females, although in some of these they are more common in females. Because they basically have 2 opportunities to get 1 dominant allele. And even with 1 dominant allele, it is enough. Now, the reason why it says probability is the same, because oftentimes if you get 2 copies of the dominant allele, it's fatal, and you don't survive. So then you have questions of, okay, well, how can we screen for these genetic illnesses? And should we? And so what we're going to do another little lecture break. The last question that we have in these slides, we're actually going to save for our final chapter. So there is one additional question, but we're not going to talk about it now because it has more to do with regulating gene expression. So what I want you to do is take a couple of minutes working with those around you, and I want you to come up with some ways in which we could screen for these genetic illnesses, and then some information about what should we do with that information. So take a couple of minutes starting now. How can we screen for them and what should we do with that information?
Well, you could take a look.
Yeah, and then like based on say the X,
You just take a look at their chromosomes. That's what I'm saying. Yeah, you screen your mom and dad to see if they have any like recessive traits, any traits at all that could potentially be passed on to the kid.
And they're talking about how like they screened like this girl's younger sister, and they predicted that she had Down syndrome. And the doctor was like,
I don't know.
what did you no. And then, yeah. And then she was born and she was like perfectly normal.
She was wrong?
sickle cell anemia, sickle cell disease.
Yeah,
You
but
probably
anyway,
see
so
a
like,
person with this kind of disease. Always for you. That's how we screen.
Oh, really?
Yeah, but what the symptoms are.
Brothers?
For example,
Yeah, no, well, till you know, so he started cracking jokes at you and then started laughing actually.
yeah, he that way you're supposed to do it. Yeah, and probably using like and so that's how we can spend much
All right. So, how do we screen for some of these genetic illnesses? You can talk about a specific genetic illness and how and when we screen, or just in generalities. So one way we can do this is even before there's even a potential offspring, right? We can do what are called karyotypes for an individual. You can have your own, you can have a karyotype done for yourself, or you can have your own genome sequenced. You can sequence your entire genome for less than $2,000. Now that in many cases is excessive. You can submit a saliva sample to 23andMe and have them screen and give you about 2 million single nucleotide polymorphisms for anywhere from $70 to $100, depending on how much they're charging at any given time. And you can use that to basically determine your propensity for particular genetic illnesses, or if you actually have a specific mutation that causes a genetic illness, if you have 2 copies of that mutation. So you could do those. That's called genetic screening, and you could do that. And a lot of times you can meet with a genetic counselor, and couples will do this if they know like a certain genetic illness is common in their family. Meet with a genetic counselor to determine the likelihood of them passing that genetic illness on to their offspring. And so that's screening even before there's even a potential offspring. And then we can screen for it in utero. And so there are multiple ways you can do that. The placenta itself is actually half embryo, half maternal tissue. So you can take some tissue from the placental wall, and again, a half of that is embryonic tissue. And so you don't actually have to take tissue directly from the embryo. Although you can, and you can screen that way. Although even just taking something from the placenta, it's hard to do that in a non-invasive way. And so a lot of the screenings that they do don't happen until after birth. And they'll take tissues from the baby at birth and basically screen for a number of these genetic illnesses. Although you can have them done in utero. Yeah, Allison.
We're just doing ultrasound and observing the phenotype of the baby.
Sure. With some, yeah, I would suggest the need for certain screenings. Yeah, Tara.
Can test it when it's in vitro fertilization?
Sure. Yeah, even before it's implanted. Absolutely. Yep. Yeah, so the way, not all animals work this way, but the way vertebrates work is you can remove a cell from even like the 8-cell stage, and both that one cell that you'd remove and the 7 remaining cells are both capable of developing into an individual. And so because of that, you'd probably want to wait till a little bit after, because now you've got some ethical concerns, right? If you remove that, you've basically created an identical twin, right? And so it's like, yeah. And that's basically what happens with identical twins, you know? So, but yes, you can screen that way before even implantation. Yeah, Davies.
Can you also, like for example, for sickle cell anemia, can you also like just take a look at the phenotype of the cells, the red blood cells, to see their shape?
Yeah. Yeah, and we're getting pretty good at imaging and looking at individual capillaries, even in utero, where you can look at individual capillaries and how the blood's flowing through and determine the shape of blood cells. Yeah. And so then the question of, well, what do we do with this information? And a lot of that's going to be governed by your particular moral and ethical framework in terms of what you do with that information. And so you have a lot of people that just choose not to do the screening because it oftentimes can't be done in a non-invasive way and increases likelihood of a miscarriage. And but again, what you do with that hopefully is governed by a biblical view of humanity, and that every individual is created in God's image, and therefore the morally responsible thing is to protect that image of God in the individual. So, all right, any questions, thoughts? Again, there's one more question to deal with here, cytoplasmic inheritance and genomic imprinting, but that fits better with the chapter that we're going to talk about next week. All right, goodbye.
Bye.